This super antibody for COVID fights off multiple coronaviruses. Scientists have uncovered an antibody that can fight off not only a wide range of SARS-CoV-2 variants, but also closely related coronaviruses. 1. The discovery could aid the quest to develop broad-ranging treatments and vaccines. Starr, a biochemist at the Fred Hutchinson Cancer Research Center in Seattle, Washington, and his co-authors set out to shed light on a problem facing antibody treatments for COVID-19. Some variants of SARS-CoV-2 have acquired mutations that enable the virus to escape the antibody's grasp. The researchers examined 12 antibodies that Vir Biotechnology, a company based in San Francisco, California, that was involved in the study, isolated from people who had been infected with either SARS-CoV-2 or its close relative SARS-CoV. Those antibodies latch onto a fragment of viral protein that binds to receptors on human cells. Many antibody therapies for SARS-CoV-2 infection grab the same protein fragment, called the receptor binding domain. The researchers compiled a list of thousands of mutations in the binding domains of multiple SARS-CoV-2 variants. They also cataloged mutations in the binding domain on dozens of SARS-CoV-2-like coronaviruses that belong to a group called the SARS-CoV. Finally, they assessed how all these mutations affect the 12 antibodies' ability to stick to the binding domain. One antibody, S2H97, stood out for its capacity to adhere to the binding domains of all the SARS-CoV that the researchers tested. S2H97, which the authors dub a pan-SARS-CoV antibody, was able to prevent a range of SARS-CoV-2 variants and other SARS-CoV from spreading among cells growing in the laboratory. It was also powerful enough to protect hamsters against SARS-CoV-2 infection. That's the coolest antibody that we described, Starr says. A closer examination of S2H97's molecular structure revealed that it targets a previously unseen and well-hidden region on the binding domain, a section that is revealed only when the domain pops up to bind to a cell's receptor. Starr notes that molecules targeting this binding domain region could generate protection against multiple viruses, and might one day be used in pan-SARS-CoV vaccines. The other 11 antibodies could target a variety of viruses, but the more effectively an antibody blocked the entry of the earliest known SARS-CoV-2 strain into a cell, the smaller the range of viruses it could bind. The team also found that antibodies that could disable a wide variety of viruses targeted sections of the binding domain that tended not to change as the virus evolved. It's good news that the team has identified antibodies that can bind to a range of SARS-CoV, says R.J. Banerjee, a virologist at the University of Saskatchewan in Saskatoon, Canada. Although scientists can't test an antibody's activity against an unknown virus, Banerjee adds, pan-SARS-CoV treatments and vaccines would help to prepare the world to fight the next coronavirus that jumps from wildlife into humans.